Let's look closer into chemical properties of the electron carriers within the electron transport chain. The first class, flavine-based carriers, represent the flavine derivatives, flavine mononucleotide or flavine adenine nucleotide, respectively FMN or FAD, which are tightly embedded in a complex with the respective carrier proteins. So the within these complexes with carrier proteins, the FMN or FAD represent parasitic groups which are either bound very strongly by non-covalent interactions or attached covalently to the respective proteins. Because of that, these electron carriers are referred to as flavoproteins. The working compound or group within the flavoproteins is the flavine group, which represents the system of unsaturated rings which could be present in oxidized quinone or reduced quinol state. The free energy difference between oxidized and reduced state is generated by various extent of electron delocalization between the higher extent of electron delocalization in the oxidized form and respectively lower extent in the reduced form, and this generates difference in free energy. Importantly, this difference and the respect and as a consequence the electron affinity is highly dependent on the surroundings, on the context in which this flavin moiety is present. So different, because the different flavor proteins generated in different context have different affinities for electrons, different redox properties. Importantly, the flower proteins, because of the properties of flavin group, can carry either 2 electrons and 2 protons, respectively corresponding to fully oxidized and fully reduced state of flavin, or 1 electron and 1 proton, corresponding to semiquinone state, which we will discuss an example of ubiquinone later. As an interesting fact, the generation of flower proteins in our body is dependent on dietary supplementation with riboflavin, which is also known as vitamin B2. So in the respective electron carriers, FMN and FAD, are generated from riboflavin through the series of small modifications. The flavin-based, flower protein-based electron carriers, as mentioned before, have the lowest affinity for electrons within the electron transport chain, and because of that, they are present in the initial steps of the electron transport chain within complexes 1 and 2. The second class of electron carriers are cytochromes. Similar to flower proteins, cytochromes are complexes between the electron carrying group and the carrier proteins. Unlike the flower proteins, the electron carrier or redox function in cytochromes is mediated not by organic electron delocalization properties, but by charge state of the iron atom present within the Hem parasitic group within the cytochromes. Within Hem, the iron could be present either in +2 reduced form or +3 oxidized form. As such, the unlike flower proteins, cytochromes could carry only single electrons and cannot carry protons. The similar to flower proteins, redox properties of cytochromes are a result of interplay between the redox properties of iron per se and the context surroundings generated by the um, polyunsaturated porphyrin ring of the hem, its chemical modifications, and the protein context within which the hem is embedded. So why cytochromes are called cytochromes? The reason is that the cytochromes are very efficiently absorbing light. And this deals with the fact that the hem represents a highly delocalized system of electrons generated by porphyrin rings, which is very efficiently absorbing the light. And the light absorption properties are highly dependent on the surroundings of this delocalized electron cloud, which is dictated by the Bose charge of the iron ion and the side chains modifying porphyrin ring and the enzyme context. And because of that, the properties of cytochromes, the redox of properties of cytochromes and the biochemical functions are highly correlated with the light absorption properties. And as a matter of fact, the initial classification, historical classification of cytochromes is based on the light absorption spectra, which could change between the oxidized and reduced forms. And this is, for example, the light absorption spectrum of soluble cytochrome C with curves showing the absorption spectrum of oxidized form and the reduced form. So the based on the spectral properties, the cytochromes are classified into three major types: A, B, and C. The A and B types are contain the hem, the parasitic group embedded non-covalently in the context of respective carrier proteins and are often modified with large organic extensions, for example, isoprenoid chain as in the case of A-type cytochromes. The A and B-type cytochromes are membrane-associated proteins and they embedded into large respiratory chain complexes, complexes 2, 3, and 4. Sorry, 3 and 4. And the cytochrome C types are covalently embedded in respective carrier proteins through the cysteine residues, and one subtype of the cytochrome C is actually soluble protein, as mentioned, which is present in the lumen between inner and outer mitochondrial membrane and used as a soluble electron carrier. The third class of electron carriers are iron-sulfur clusters within the electron transport chain proteins. The similar to flower proteins and the cytochromes, these electron carriers are embedded within the context of carrying proteins, and they redox or electron affinity properties are mediated by the charge state of the iron atom embedded into the context of proteins. Unlike the porphyrin rings in cytochromes, the iron atom in iron sulfur clusters is embedded by binding to sulfur provided by cysteines within the respective carrying proteins, and the iron sulfuric acid can come in different flavors, but contain the um, equal number of iron and sulfur atoms. In the last class to discuss is well, standalone class with containing one factor, ubiquinone, which is also known as coenzyme Q. So ubiquinone is a small molecule, unlike other types of electron transport carrier. It's not embedded in the context of carrying proteins, but is a free-floating molecule within the inner mitochondrial membrane due to its highly hydrophobic properties. Ubiquinone represents the benzoquinone group with attached the long isoprenoid chain, which altogether gives rise to highly hydrophobic properties and exquisite ubiquitous membrane solubility. So the benzoquinone group is gaining its electron affinity redox properties due to various extent of electron delocalization, similar in a sense to flavin-based electron carriers. In the fully oxidized quinone state, the electrons within the benzoquinone head are least delocalized, and the ubiquinone can act as a versatile electron acceptor, which could accept either one proton or one electron, converted into a semiquinone radical state, or accept two protons and two electrons, converted to fully reduced ubiquinol state. And as we will see later, it will become very practical during certain steps of the electron transfer within the electron transfer chain. By being a small membrane soluble molecule, the ubiquinone is play an important role in carrying the electrons within the electron transfer chain in long distances within the inner mitochondrial membrane, transferring them between the gateway of the electron transport chain complexes 1 and 2 to the further steps of the electron transport according within complex 3.